Hey everyone, welcome back to the channel MLT Lectures. If you are new to this channel, here we discuss everything related to medical laboratory course. So don't forget to subscribe the channel. In this video, we will discuss about hemoglobin and its estimation by various methods in the laboratory. The objective of this video is to know about the basic structure of hemoglobin, function of hemoglobin, various laboratory methods for estimation of hemoglobin, and lastly enumerate the advantage and disadvantage of each method. Now let's move on to the introduction part of hemoglobin. Hemoglobin is a major constituent of the red cell cytoplasm, accounting for approximately 90% of the dry weight of the mature cell. It is comprised of heme and globin. Now, moving to structure of hemoglobin, it is a tetramer consisting of two pairs of similar polypeptide chain called globin chains. To each of the four chains is attached with the heme, which is the complex of iron in a ferrous form and protoporphyrin. The major type of hemoglobin present in adult normal human being is HbA, which consists of 2 alpha globin chains and 2 beta globin chains. Here in the figure we can see a hemoglobin structure consisting of 2 alpha chains and 2 beta chains, along with the heme group. The heme group contains iron in the ferrous state. The gene that codes for formation of alpha globin is located on chromosome 16, whereas the gene that codes for formation of beta globin chain is on chromosome 11. A minor amount of HbA2 is also present in normal human adults. Now, function of hemoglobin. The heme has the ability to bind with oxygen and it carries oxygen to the tissues. It also facilitates the exchange of carbon dioxide between lungs and tissue. Thus, hemoglobin functions as the primary medium of exchange of oxygen and carbon dioxide. The normal concentration of hemoglobin in humans varies. In men, it is 15 to 20 gram per liter. In women non-pregnant, 13.5 to 15 gram per liter. In pregnant women, during first trimester, it is 12.4 to 13.5 gram per liter. In second trimester, it is 11 to 11.7 gram per liter. In third trimester, it is 10.6 to 10.9 gram per liter. During the birth, the hemoglobin concentration is maximum. It is about 18 to 23 gram per liter. Now, physiological variation in hemoglobin concentration. During renal insufficiency, inflammation, testosterone deficiency, diminished erythropoiesis, we can see reduction in hemoglobin concentration. Whereas during exercise, altitude, smoking, there is increase in hemoglobin concentration. Now, when is hemoglobin determined or indication of hemoglobin determination? It is used to determine presence and severity of anemia. It is used for screening for polycythemia, to detect the response to the specific therapy in anemia, to estimate red cell indices, and for selection of blood donors. Now, various methods to determine hemoglobin include: firstly, by colorimetric method. It includes Sali's acid hematin method, Tallquist hemoglobin chart, WHO hemoglobin color scale, oxyhemoglobin method, specific gravity method. Photoelectric method includes Sali hemoglobin method, oxyhemoglobin method, and alkaline hematin method. Now, firstly, we will discuss about gasometric method. Here, the oxygen carrying capacity is measured by using Van Slyke apparatus. Based on the formula, 1 gram of hemoglobin carries 1.34 ml of oxygen. The calculation is done. This method does not measure carboxyhemoglobin, sulfhemoglobin, methemoglobin. This method is time consuming and expensive. Result is 2% less than other methods. Chemical method. Here, iron content of the hemoglobin is first estimated. Then, indirectly hemoglobin is derived. 100 grams of hemoglobin contains 374 grams of iron. This method is also time-consuming method. This method is used to calibrate all other methods of hemoglobin estimation. Next method we have is specific gravity method. Rough estimate is made from specific gravity of blood. Here, copper sulfate is used. So this method is also called as copper sulfate technique. It is used in the mass screening like selection of donor. It is rapid and simple method, commonly used in blood donor selection. Procedure of specific gravity method. A drop of blood is allowed to fall in a copper sulfate solution of specific gravity of 1.053 from a height of 1 cm. It is considered that the specific gravity is equivalent to 12.5 grams deciliter. If a drop gets covered with the copper proteinate, if the drop sinks, specific gravity is higher than the copper sulfate, so the hemoglobin is more than 12.5 grams per liter. Here we can see a copper sulfate solution and a lancet where a finger is pricked and a drop of blood is allowed to sink or float according to the Next we have is Sali's acid hematin method. Principle of Sali's acid hematin method. The blood is mixed with an acid solution so that Hb is converted to brown colored acid hematin, then diluted with water till the brown color matches the brown glass of the standard. The Hb value is directly read from the scale. Equipment required for determination of hemoglobin by Sali's hemoglobin method are Sali's hemoglobinometer, Sali's pipette, stirrer, and dropper pipette. Reagents used are 1/10 and hydrochloric acid and distilled water. This is the diagram of Sali's hemoglobinometer. Procedure: place 1/10 NaCl into a HB tube up to 2 grams. Blood sample in the Sali's hemoglobin pipette up to 20 microliter. Then add blood sample to the acid hematin solution. Mix with stirrer. Allow to stand for 10 minutes. Add distilled water drop by drop till the color of the solution matches to the brown glass standard. Take the reading of the lower meniscus from the graduated tube. Advantage: easy to perform, quick, inexpensive, can be used as a bedside procedure, does not require technical expertise. Disadvantage of this method is for maximum color, longer time is required. Perfect matching with the brown glass standard is not possible. Carboxyhemoglobin, methemoglobin, and sulfhemoglobin are not converted to acid hematin. Development of a color is slow and acid hematin is not stable. Source of light will influence the comparison of colors. Next method we have is Cyanmethemoglobin method. Most accurate method for estimation of hemoglobin. This method is recommended by International Committee for Standardization in Hematology because in this method all forms of hemoglobin are converted into Cyanmethemoglobin with the exception of sulfhemoglobin. This method is stable and reliable standard in principle of Cyanmethemoglobin method. Here, blood is mixed with the Drapegreen solution. Erythrocytes are lysed, producing an evenly distributed hemoglobin solution. After that, potassium ferricyanate converts hemoglobin to methemoglobin. Then, methemoglobin combines with potassium cyanide to form cyanmethemoglobin. As all hemoglobins present in the blood are converted to this form, that is cyanmethemoglobin, absorption is then measured at the spectrophotometer at 540 nanometer. To obtain amount of unknown hemoglobin sample, each observance is compared with the standard cyanmethemoglobin solution. Drapegreen's solution has a pH of 7.0 to 7.4. It contains potassium ferricyanide, potassium cyanide, potassium dihydrogen phosphate, non-iodinated detergent, and distilled water. For this method, we need photoelectric colorimeter or spectrophotometer, Sali's pipette of 20 microliter and pipette of 5 ml. Procedure: take 5 ml of Drapegreen solution and add 20 microliter of blood. Use a stopper and mix by inverting several times. Allow it to stand for 5 minutes. Transfer the sample to the cuvette. Then read the observance in the spectrophotometer at 540 nanometer filter. Also take the observance of standard solution. This is the formula to calculate hemoglobin by Sali method. Hemoglobin in gram per dl is equal to observance of test sample upon observance of standard into concentration of standard multiplied by dilution factor upon 100. Advantages of Cyanmethemoglobin method are as follows. All forms of hemoglobin except sulfhemoglobin are converted to Cyanmethemoglobin. Visual error is not there as no color matching is required. Cyanmethemoglobin solution is stable and its color does not fade with the time, so reading may not be taken immediately. Observance may be measured soon after dilution. A reliable and stable reference standard is available from WHO for direct comparison. Disadvantage: diluted blood has to stand for a period of time to ensure complete conversion of hemoglobin. Prolonging the reaction time to 30 minutes can overcome this problem. The rate of conversion of blood containing carbohemoglobin is slowed considerably. Potassium cyanide is poisonous substance, and that is why drops in solution must not be never prepared by mouth. Abnormal plasma proteins causes turbidity when the blood is diluted with the Tramgren solution. Now, hemoglobin estimation by automated blood count method. This is the modification of Cyanmethemoglobin method. Here, other chemicals like sodium lauryl sulfate, imidazole, sodium dodecyl sulfate can be used. Measurements are made at various wavelengths depending on final stable product. Next we have is Thalquist hemoglobin chart method. Here, series of lithographed colors said to be correspond to the HV values ranging from 10 to 100% is available. Blood is obtained from the finger puncture, then placed on a piece of absorbent paper. Then the color is matched against the color on the chart. Corresponding reading is taken. This is cheap and simple method, but the disadvantage is error can take place from 20 to 50%. Next we have is hemoglobin color scale, that is WHO hemoglobin color scale. This was devised by Scott and Lewis. The principle is similar to that of Thalquist method. It is rapid, simple, inexpensive and reliable method. This is used for diagnosis of anemia. Printed set of color corresponding to hemoglobin varies from 4 to 14 gram per deciliter. 86% in classifying to its grade WHO hemoglobin scale. It is useful for screening blood donors and the iron therapy. This is the WHO hemoglobin color scale with the blood sample. Thank you. Hope you like the content. Please like, share and subscribe the channel.